Rok, wstawiłeś już tą pierwszą anatomię?
Nie, tego jeszcze nie.
Nie?
Nie miałam siły.
To co ty robiłeś w domu?
Siedziałam po prostu, się relaksowałem. Tak, mi się wydaje. Te pliki trzeba...
Sam tak stwierdziłeś.
Ale wiesz co? Jak weźmy jeszcze pliki, to żeby rozmiar odpowiednio jest wielki, chyba godzina 15 minut. Ja to muszę złożyć, złożyć podpisy, wyciąć tam, gdzie widać, co nagrywa, wiesz, coś tam. Trochę zajmuje. A, nie chciałem. Co mam jakiś test? A, fu.
Nie idę spać tutaj, nie wiem. Zobaczcie, patrz nogę, dopiero to trzymam.
Uśpiony?
A wtedy on mówi, że to jest
Co, jak pomyślisz, wielu liczaliśmy dla fizyki, czy innych udało się?
no nie wiem, że to
Samo, jesteśmy raz. To znaczy, że my z Polski mamy lepsze predyspozycje do tego. No, jasne.
Krishan, come here real quick, please. Come here real quick, please. Come here. No. Biochemistry, health cycle. You answered it out loud. Teaching examination.
What's there? That's same. That one is same. So yeah, if you throw it out, it's better, but you should know that. I should know this. What is the
I don't care.
They're gonna fight.
I don't care. He can die for all I care. Seriously.
No, I can't.
You loved her. I loved him.
No, Rachel, I was we should be that one.
that you're a girl? So this is
But you're supposed to. You're supposed to. I mean, unless it's not, that would make what, yeah. How much stuff do you need to see?
Ei, mä laitan sen niitä happuja tonne Suomeen. Meinaatko käydä? Okei, oot alkanut harjoittelemaan vielä. Mitä muuten meni?
Okay, so at the beginning, I would like to remind you that the next lecture will be on the next Monday. So you don't have classes on next Monday, but you have lecture. So let's meet next Monday at the same time. Okay, so now I'm going to continue my talk on hormone receptors. So as you already know, all hormones have their target cells. You already know how the insulin works. So as you remember, insulin interacts with membrane receptor and it stimulates phosphorylation of several proteins within the cell. But most of hormones which interact with membrane receptor employ so-called second messengers. Second messenger. Please remember that most of hormones interact with cell membrane with participation of secondary messenger. For instance, this is resting state. Hormone, receptor, protein G, and inactive amyloid cycles. This is extracellular space. This is cytosol surface of cell membrane. So this is cell membrane constructed with phospholipids, mainly hormone receptor, protein G, and inactive adenine cyclase. Adenine cyclase is an enzyme. Protein G is a membrane protein which participates in hormone receptor interaction. So as you see, the G protein is composed of 3 subunits: alpha subunit, beta subunit, and gamma subunit. The alpha subunit binds GDP. GDP is guanosine diphosphate. Guanosine diphosphate. This is in resting state. What happens next? In the next step, the receptor binds hormone. The receptor which bound a hormone interacts with G protein. G protein, alpha subunit, beta subunit, gamma subunit, and alpha subunit binds GDP. The adenine cyclase does not participate in this reaction. What happens in the next step? The receptor hormone protein G complex changes GDP for GDP. So GDP, guanosine diphosphate, is replaced by GDP, guanosine triphosphate. GDP is released. What happens next? In the next step, the protein G dissociates. It separates from the receptor hormone complex. Alpha subunit is released, and it interacts with adenine cyclase. So the binding of alpha subunit with GDP results in activation of adenine cyclase. So the inactive adenine cyclase is converted into active adenine cyclase. What happens next? In the next step, the adenine cyclase interacts with ATP. It converts the ATP into cyclic ATP and inorganic pyrophosphate. What happens next? In the next step, GDP is hydrolyzed, inorganic phosphate is released, GDP is converted into GDP. The alpha subunit dissociates from adenine cyclase. It interacts again with beta subunit, and hormone separates from receptor. The active adenine cyclase is converted into inactive adenine cyclase. This is how the hormone interacts with its receptor. This is very important, what I said. Please look again. This is membrane, cell membrane. These are phospholipids. These are protein components of cell membrane. This is hormone receptor. This is protein G. This is inactive adenyl cyclase. This is GDT. So in resting state, the alpha subunit binds GDT. What happens after binding of hormone to receptor? So the complex hormone receptor interacts with protein G. It associates with protein G. GDT is replaced by GDT. In the next step, the alpha subunit dissociates from protein G and associates with adenyl cyclase. Inactive adenyl cyclase becomes active adenyl cyclase. What happens next? In the next step, the active adenyl cyclase interacts with ATP. ATP is converted into cyclic AMP with a release of inorganic pyrophosphate. In the next step, GDT is hydrolyzed, inorganic phosphate is released, and GTP is replaced by GDT. Hormone dissociates from its receptor. So as you see, the alpha subunit has GTPase activity. It converts GTP into GDT. What is the mechanism of action of adenyl cyclase? So adenyl cyclase converts ATP into cyclic AMP, cyclic 3' 5' AMP. This is adenine, this is ribose, phosphate, phosphate, and phosphate. This is ATP. So under the action of adenyl cyclase, this fragment of molecule is split off in a form of inorganic phosphate, and this phosphate residue interacts with OH group and carbon 3' of ribose, forming cyclic AMP, cyclic 3' 5' AMP. So this phosphate group forms two ester bonds with OH prime and carbon 3' and with OH group and carbon 5'. This is cyclic AMP. What is the biological role of cyclic AMP? Cyclic AMP activates protein kinase A. This write down and remember what is the biological role of cyclic AMP. Cyclic AMP activates protein kinase A. Let's look at the mechanism of this reaction. This is protein kinase A. So protein kinase A is an enzymatic protein composed of 4 subunits. It contains 2 regulatory subunits and 2 catalytic subunits. In resting state, the complex of regulatory subunits and catalytic subunits is inactive. The catalytic subunits contain acceptors for cyclic AMP. This one, this one, this one, and this one. So one molecule of inactive protein kinase A binds 4 molecules of cyclic AMP. So the binding of cyclic AMP by regulatory subunits change the shape of regulatory subunits. They dissociate from catalytic subunits. So in a complex, the catalytic subunits are inactive. After dissociation from regulatory subunits, they become active. So in resting state, the regulatory subunits exert an inhibitory action on catalytic subunits. After binding of cyclic AMP, the catalytic subunits dissociate from regulatory subunits and they become active. What does the catalytic subunits of protein kinase A? So the catalytic subunits phosphorylate various proteins. In some cases, the phosphorylation of enzyme results in activation. In some cases, the binding of phosphate results in inactivation of an enzyme. How the cyclic AMP is degraded? So this is cyclic AMP. Cyclic AMP is degraded by phosphodiesterase. So cyclic AMP, under the action of water molecule and the enzyme phosphodiesterase, is cleaved at this point. This ester bond is cleaved. OH group appears at carbon 3'. Cyclic AMP is converted into AMP. This molecule is active. This molecule is inactive. There are some substances which inhibit phosphodiesterase. For instance, coffee and theophylline. This is coffee. This is theophylline. So as you see, the coffee is similar to purine bases existing in nucleotides and nucleic acid. So coffee contains 3 methyl groups. 1, 2, 3. And 2 CO groups. 1, 2. The theophylline is very similar. It does not contain this methyl group and this nitrogen. Everything else is identical. Coffee and theophylline are components of coffee and tea. Therefore, drinking coffee or tea results in inhibition of phosphodiesterase and prolongation the effects exerted by hormones. What are the effects of action of protein kinase A? So protein kinase A phosphorylates enzymatic proteins, increasing or decreasing their catalytic activities. Protein kinase A phosphorylates histones and ribosomal proteins, increasing or decreasing transcription or translation processes. This is very important. Please write down and remember. This is a mechanism of action of protein kinase A on intracellular proteins. This is an example of the action of protein A on an enzymatic protein. So you already know that there is an enzyme which is called glycogen phosphorylase. Glycogen phosphorylase is an enzyme engaged in glycogen degradation in liver and in muscles. In the resting state, the phosphorylase, glycogen phosphorylase, exists in a form B. Under the action of protein kinase, 2 molecules of glycogen phosphorylase B interact with 4 molecules of ATP. 2 molecules of phosphorylase B are converted into 1 molecule of phosphorylase A. 4 molecules of ATP are produced. So please look again. This is inactive enzyme. After phosphorylation with ATP, it is converted into active enzyme. Phosphorylase B is converted into phosphorylase A. So you already know how the protein A is activated. And now let's look how the protein kinase A is inactivated. So degradation of site 3' 5' AMP by phosphodiesterase results in activation of protein phosphatases, which dephosphorylate proteins, converting them into previous form. So there are specific enzymes, protein phosphatases, which cleave off phosphate groups from amino acids. 5' AMP, product of 3' 5' AMP degradation, dissociates from protein kinase A, converting this enzyme into inactive form. So, dephosphorylated subunits associate, forming inactive complex. The effects exerted by hormone are cancelled by the action of protein phosphatase. Which hormones act through cycling 3' 5' AMP? First of all, epinephrine, norepinephrine, glucagon, calcitonin, parathormone, vasopressin, melanotropin, chorion, and ACTH, FSH, and LH. ACTH is adrenocorticotropic hormone. Adrenocorticotropic hormone. FSH is follicle stimulating hormone. LH is luteinizing hormone. These hormones interact with its receptor. Receptor interacts with protein G. The alpha subunit of protein G binds GTP with a release of GTP. The complex of alpha subunit with GTP activates adenyl cyclase. Activated adenyl cyclase converts ATP into cyclic AMP. Cyclic AMP activates protein kinase A. Protein kinase A activates or inactivates intracellular enzymatic proteins. Some hormones act on a cell with participation of cyclic GMP, cyclic 3' 5' GMP. So, in contrast to cyclic KT, the cyclic GMP activates protein G. Protein kinase G. In contrast to cyclic 3' 5' AC, it participates in smooth mass relaxation, blood platelet aggregation, and visual stimuli transduction. So please remember that cyclic AMP participates in the regulation of enzymatic activity, and cyclic GMP participates in smooth mass relaxation, blood platelet aggregation, and visual stimuli transduction. Some enzymes act on cell with participation of deacylglycerol, and inositol 1,4,5,3-phosphate. The abbreviation of deacylglycerol is DAG. The abbreviation of inositol 1,4,5,3-phosphate is IP3. So some hormones interact with phospholipase C. Phospholipase C interacts with membrane phospholipids, mainly with phosphatidyl inositol 4,5-bisphosphate. Phosphatidyl inositol 4,5-bisphosphate. So this is phosphatidic acid, this fragment, and this is 4,5 inositol bisphosphate. Phosphatidic acid, inositol, one phosphate group, second phosphate group. The third phosphate group belongs to phosphatidyl inositol. So under the action of phospholipase C, this bond, this ester bond is cleaved with participation of water molecule. Phosphatidyl inositol 4,5-bisphosphate is converted into deacylglycerol, DAG, and inositol 1,4,5-bisphosphate, IP3. This is a very important reaction. You have to remember this reaction. Both DAG and IP3 are segment messengers. How DHA and IP3 are degraded. So, IP3 is degraded by dephosphorylation. So, under the action of water, 3 molecules of water, these phosphates are split off. They are released in the form of inorganic phosphate. IP3 is converted into inositol. IP3 is active, inositol is inactive. How DHA is degraded. So DHA is degraded with participation of phospholipase A2. A2. So phospholipase A2 with participation of water molecule cleaves off this fatty acid at carbon 2, converting the deacylglycerol into monoacylglycerol. The deacylglycerol is inactive as second messenger. The monoacylglycerol is inactive. How phosphatidyl inositol 4,5 bisphosphate is synthesized. So phosphatidyl inositol 4,5 bisphosphate is synthesized from phosphatidyl inositol. Phosphatidyl inositol is a component of cell membrane, phospholipid component of cell membrane. It is phosphorylated by ATP. This OH group and this OH group bind phosphate residue from ATP, converting into phosphatidyl inositol 4,5 bisphosphate. 2 molecules of ATP are consumed in this reaction. What are the functions of second messengers, IP3 and DHA? So, IP3 diffuses from the cell membrane. Please remember that phosphatidyl inositol and phosphatidyl inositol 4,5 bisphosphate are components of cell membrane. So, IP3 diffuses from the cell membrane to cytoplasm and binds to its receptor in reticuloendoplasmatic membrane. Very important statement, please remember, IP3 diffuses from the cell membrane to cytoplasm and binds to its receptors in reticuloendoplasmatic membrane. IP3 binding results in a release of calcium ions from reticuloendoplasmatic cisterns, giving a rapid increase in calcium ion concentration in the cytosol. The calcium ion dependent processes are enhanced. This is very important statement. Please remember that IP3 modifies the distribution of calcium ions within the cell. And now, let's look how the other secondary messenger, DAG, works. So, DAG remains the cell membrane. So please remember that IP3 diffuses from cell membrane to cytoplasm, whereas DAG stays in the membrane. It activates protein kinase C, which phosphorylates specific serine and threonine residues in various proteins. So as you see, the phosphorylase, the protein kinase C exerts similar reactions like protein kinase A. Some hormones, like epinephrine, act on the cell with participation of two hormones. Please ignore Polish names. This is cell membrane. This is alpha adrenergic receptor of epinephrine. This is epinephrine. This is receptor. Adrenaline is epinephrine. So epinephrine may interact with a cell through alpha adrenergic receptor and beta adrenergic receptor. So, the binding of adrenaline, of epinephrine with beta adrenergic receptor results in activation of adenine cyclase. Adenine cyclase converts ATP into cyclic A. The binding of epinephrine by alpha adrenergic receptor activates phospholipase C. Phospholipase C converts phosphatidylate inositol into DAG and IP3. So as you see, the same hormone may exert different effects. It depends on the type of receptor. For instance, epinephrine has two receptors. Alpha adrenergic receptor and beta adrenergic receptor. Alpha adrenergic receptor activates phospholipase C. Beta adrenergic receptor activates adenine cyclase. The next. Second messenger is calcium ion. Calcium ion. Many signals accepted by cell result in a change in calcium ion concentration in cytosol. Such an alteration is observed mainly in the master cells. So please remember that the contraction and relaxation of skeletal muscles are accompanied by a change of calcium ion concentration within different compartments of cells. So the concentration of calcium ion is in cytosol is low. It's about 0.1 micromole per liter. It is 10,000 times lower than in extracellular fluid and 1,000 times lower than in sarcoplasmatic system. Muscle contraction is induced by a rapid increase of calcium ion concentration in cytosol, whereas muscle relaxation is induced by a rapid removal of calcium ions from cytosol. The next, second messenger is nitrogen oxide. So nitrogen oxide is a product of arginine metabolism. Let's look at these reactions. This is arginine, carboxylic group, amino group, and guanine group here. It is under the action of nitrogen of NO synthase, with participation of NADPH plus H plus. It is hydroxylated in this place, at this point. And ADPH is converted into NADP. One oxygen interacts with these two hydrogens, forming water molecules. And one oxygen atom is incorporated in this point, forming hydroxyl group. The arginine is converted into hydroxyarginine. The reaction is catalyzed by NO synthase. The next step is also catalyzed by NO synthase, with participation of oxygen and NADPH plus H plus. One oxygen interacts with two hydrogen atoms, forming water molecule. The other oxygen interacts with this nitrogen, forming nitrate oxide, nitrogen oxide. The hydroxyarginine is converted into citrulline. So the NO synthase catalyzes two-step reaction. In the first step, the L-arginine is converted into hydroxyarginine, and ATPH plus H plus is consumed, converted into NADP plus. Oxygen molecule is divided into two parts. One atom incorporates into hydroxyarginine, the other atom interacts with hydrogen or with water. In the next step, hydroxyarginine is converted into citrulline, and O molecule is released. Hydroxyarginine is converted into citrulline. Next molecule of NADPH plus H plus is consumed, and one oxygen molecule is consumed. So NO, nitrogen oxide, also serves as second messenger. Here are listed the main functions of nitrogen oxide. So nitrogen oxide induces the relaxation of smooth mass, especially in vascular work, resulting in a decrease of arterial pressure. It works as a modulator, as a neuromodulator in central nervous system. It inhibits blood platelet aggregation. It stimulates phagocytic function of macrophages. NO is very unstable. It exists for 6 to 8 seconds. It is rapidly oxidized, forming nitrate 3 and nitrate 5. So these are the main functions of nitrogen oxide as a second messenger. What is the biological significance of second messengers? So, secondary messengers make possible amplification of hormonal stimuli. Secondary messengers make possible amplification of hormonal stimuli. For instance, one molecule of epinephrine reacts with one membrane receptor, resulting in formation of several hundreds of cytokine molecules. One molecule of epinephrine reacts with one membrane receptor, resulting in formation of several hundreds of cytokine molecules. Each 4 molecules of cytokine B activate one molecule of protein kinase. Not C but A, but with one molecule of protein kinase A, not C but A. Each molecule of this enzyme phosphorylates from hundreds to thousands of other protein molecules. This is the biological sense of existing of secondary messages. So you already know how the membrane receptors of hormones function in a cell stimulated by a hormone. And now let's talk about cytosol and nuclear receptors of some hormones. So steroid hormones and thyroid hormones have intracellular receptors. Not membrane receptors, but cytosol or nuclear receptors. So some hormones are hydrophobic. They penetrate through the cell membrane and interact with intracellular receptors. The receptors of glucocorticoids and mineralocorticoids are localized in cytosol, whereas the receptors of other steroid hormones and thyroid hormones are localized in nucleus. Very important statement. The cytosolic receptor is a protein which contains three functional domains. C-terminal hormone binding domain, central DNA binding domain, and N-terminal domain, which conditions transcription activity of the receptor. Also very important information, please remember that cytosolic receptor has three different functional domains. One which binds hormone, one which binds DNA, and the third one which conditions transcriptional activity of the receptor. The DNA binding domain of resting receptor is unable to bind DNA because it is associated with heat shock protein of the molecular weight 90 kilodaltons. The abbreviation of the name is HSP. Heat shock protein 90. Heat shock protein 90. HSP 90. Hormone binding by C-terminal domain causes dissociation of HSP19 from the receptor and makes it able to penetrate the nuclear membrane and to bind to specific sequences of DNA, which is referred to as hormone response element. Hormone response element. Hormone response substance, or you may use the abbreviation HRE or HRC. LIS. The HRS sequence is localized in the vicinity of G, which is regulated by individual hormone, usually several hundred bases upstream from the place of transcription initiation. What means upstream? Upstream means in the direction of 5' terminus of DNA. The hormone receptor complex, which is bound to DNA, plays a role of transcription factor. It facilitates association of RNA polymerase with DNA and promotes or inhibits transcription of a gene. Product of transcription, mRNA, translocates to cytoplasm. It is submitted to translation, resulting in synthesis of a protein encoded by a gene. This is a very difficult process. Please write down and think a moment on this process. So the receptor of glucocorticoid, of the mineralocorticoid, is localized in cytosol. After binding a hormone, the heat shock protein is released, and hormone receptor complex becomes able to penetrate the nuclear membrane and interact with DNA. Here is an example of stimulatory action of glucocorticoid receptor complex. So, glucocorticoids bind to their cytosol receptor. The hormone receptor complex translocates to nucleus. It binds to DNA, stimulating mRNA synthesis. The mRNA migrates to cytoplasm and it is submitted to translation process, resulting in synthesis of gluconeogenesis enzymes, pyruvate carboxylase, phosphonol, pyruvate carboxykinase, and glucose 6-phosphatase. So, as you remember, these three enzymes are the key enzymes in gluconeogenesis process. Their biosynthesis is stimulated by glucocorticoids. And now, let's look. Now let's look at the inhibitory action of glucocorticoid receptor complex. So in some cases, the glucocorticoid receptor complex exerts inhibitory effect on transcription process. E.g., for instance, the transcription of pro-opioid melanocortin gene is inhibited. Pro-opioid melanocortin is a precursor of 8 peptide hormones, including ACTH, adrenocorticotropic hormone. Such a phenomenon makes possible to regulate glucocorticoid synthesis. So ACTH is a pituitary gland hormone which stimulates glucocorticoid production in the adrenal gland cortex. When plasma concentration of glucocorticoids reaches optimal value, they exert inhibitory effect on ACTH production. It results in inhibition of further glucocorticoid synthesis. Please write down and remember. Another example of inhibitory action of glucocorticoid receptor complex. Glucocorticoid receptor complex inhibits the expression of cyclooxygenase gene. Cyclooxygenase is an enzyme which participates in prostaglandin synthesis. Prostaglandins are mediators of inflammatory processes. The action of glucocorticoid receptor complex decreases prostaglandin production and inhibits the inflammatory process. This is what you should know about hormones, about the biological effects of hormones, and about the mechanism of hormone receptor interaction. And now, let's talk about porphyrins. What are porphyrins? So porphyrins are natural pigments. They are composed of 4 pyrrole rings held together with metal bridges. This is general structure of porphyrin. It has 4 pyrrole rings, A, D, C, and D. all of them are held together with CH groups. 1, 2, 3, 4. The rings contain double bonds. The position of 1, 2, 3, 4, 5, 6, 7, 8 are occupied by subsequent, by methyl groups, by protonate, and by acetate and dinyl groups. The most common porphyrin is heme. So heme is a component of hemoglobin, myoglobin, and some respiratory enzymes located in mitochondria. This is general, this is a chemical structure of heme. The position 1, 3, 5, and 8 are occupied by methyl groups. One, 3, 5, 8. The position 2 and 4 are occupied by dinyl groups. 1, 2. The position 6 and 7 are occupied by propionate. So one may say that heme is 1, 3, 5, 8 tetramethyl, 2, 4 dinyl, 6, 7 dipropionate, protoporphyrin 9. Protoporphyrin 9. The central part of molecule is occupied by ions. Iron 2 plus. In hemoglobin and myoglobin, the only form of iron existing in these pigments are ions Fe2 plus. In mitochondrial, the iron exists in a form of 2 plus or 3 plus. In hemoglobin, one valency of iron is occupied by oxygen, one with histidine, which belongs to protein. The iron forms two covalent bonds with two nitrogen atoms of pyrrole rings, and two other coordinate valences are located between iron and nitrogen of other two rings. This is heme, existing in hemoglobin and myoglobin. The other porphyrin compounds is chlorophyll. So chlorophyll is a green pigment of plants. It is engaged in photosynthesis process. So it contains typical porphyrin rings with four methyl group at position 1, 3, 5, and 8. Two vinyl groups at position 3 and 4. It contains here additional cyclic structure and propionate located in the target cell forms an ester bond with phytol. Phytol is not shaped alcohol. The central part of molecule occupies magnesium. The magnesium forms two covalent bonds with nitrogen atoms and two coordinate bonds with other. Nitrogen atoms. The third, biologically active, biologically important porphyrin compound is vitamin 12. Vitamin 12. So vitamin 12 also combines for five pyrrole rings. 1, 2, 3, 4, and several subsequent in distal positions of porphyrin rings. Furthermore, it contains something like a nucleotide, which is called ribosome. Methyladenine, ribophosphate. The central position in vitamin 12 molecule which usually is methylated. Vitamin B12 is engaged in red blood cell formation. Now, let's talk about the biosynthesis. Heme. So heme is synthesized from succinyl-triazine in glycine. This is succinyl-triazine. This is glycine. Under the action of beta-glucuronidase synthase, the coenzyme A, whose SH2 is cleaved off, and then rest. The succinyl group interacts with this carbon, forming alpha-amino beta-keto ID payload. Alpha-amino beta-keto ID payload. This fragment originates from glycine. This fragment originates from succinyl-triazine A. In the next step, the alpha-amino beta-keto ID is decarboxylated. This fragment of molecule is split off in form of carbon dioxide, and the rest of molecule is converted into delta-aminoregulase. What happens next? In the next step, one molecule of alpha-aminoregulase, of delta-aminoregulase, interacts with another molecule of delta-aminoregulase. Under the action of delta-aminoregulase degradase, two molecules of water are released. The delta-aminoregulase is converted into porphyrinogen. Pair ring, acetate group, and methyl amino, methyl group, and A group, CH, C, and propionate. Acetate, propionate, amino, methyl group. This is porphyrinogen. What happens next with porphyrinogen? Four molecules of porphyrinogen interact with signal cells, forming uroporphyrinogen 3. Uroporphyrinogen 3. Four molecules of ammonia is released. The reaction is catalyzed by uroporphyrinogen synthase. Now please write down your names on these pieces of paper.